All right, welcome to your rapid-fire review of cell biology. Got a big exam breathing down your neck? Well, you're in exactly the right place. We're going to break down all the core concepts you absolutely, positively need to know.
Okay, no time to waste, let's dive right in. We're going to focus on the key stuff, the stuff that's actually going to be on the test, especially when it comes to the eukaryotic cell. This is all about giving you that confidence boost right before you walk into the exam room.
Okay, first up, the two types of cells. This is a fundamental divide, and honestly, if you don't get this, everything else gets way more confusing, so let's lock it in.
Okay, check this out. On the left, you've got your prokaryote. Think bacteria. Super simple, right? Its DNA is just kind of floating around in this area called the nucleoid. But then look at the right. That's a eukaryote. That's us, plants, animals, fungi. See all those little compartments? It's way more complex, and each one of those little rooms is an organelle with a very specific job.
And here's the cheat sheet. This table really boils it all down. If you remember one thing for your exam, make it this. Eukaryotes have a true nucleus and all those other membrane-bound organelles. Prokaryotes? Nope, they don't. That, plus the fact that eukaryotes are way bigger, and you've nailed the core difference.
All right, now for the fun part. Let's take a tour inside that complex eukaryotic cell. And to make sense of all those parts, we're going to use a really helpful analogy.
I want you to picture the cell as this tiny, super busy, and incredibly organized factory. Seriously, it's got to make stuff, power itself up, ship things out, and handle all the trash. Every single organelle is like its own specialized department, and they all have to work together perfectly to keep the whole factory running.
So let's meet the factory crew. The nucleus? That's the CEO's office. It holds the DNA, which are the master blueprints for everything. The ribosomes are the actual factory workers on the floor, building the products, the proteins. The endoplasmic reticulum is the assembly line. The Golgi apparatus, that's your packaging and shipping department. Lysosomes are the cleanup crew, handling waste disposal and recycling. And the mitochondria, well, they're the power plants, keeping the lights on for the whole operation.
Okay, let's see this factory in action. We're going to follow a single product, a protein, all the way from the blueprint to final delivery. Pay attention here, because this is a classic exam topic.
And our journey starts right here, on the main assembly line. This is the rough endoplasmic reticulum, or rough ER. Why rough? See all those little dots? Those are the ribosomes, our factory workers stuck all over it. This is where any protein that needs to be shipped out of the cell gets made.
So, as the ribosome builds the protein chain, it gets fed right into the ER. And inside, two crucial things happen. It folds up into its proper 3D shape, and it gets tagged. Think of it like a chemical shipping label that says exactly where this protein needs to go.
Okay, pop quiz time. Our protein is built, but it's not quite finished. It needs a few tweaks and has to be packaged up for shipping. So, which department in our factory is going to handle that? Think about it.
You got it. It's the Golgi apparatus, sometimes called the Golgi complex. This is the cell's post office and finishing center, all in one. It takes in the rough products from the ER, puts on the finishing touches, sorts them, and then packages them up for their final delivery.
So let's trace the whole path, step by step. Step 1, it's made on the rough ER. Step 2, it buds off in a little transport bubble called a vesicle and travels to the receiving dock of the Golgi, the cisface. Step 3 and 4, it works its way through the Golgi stacks, getting modified and sorted along the way. And finally, step 5, it's packaged into a brand new vesicle at the shipping dock, the transface, and sent out of the cell. That final exit process is called exocytosis.
And this picture puts it all together. You can see the whole flow, from the rough ER to the Golgi and then out of the cell. This whole process is called vesicular trafficking, and it's basically the cell's internal logistics system. This network, the ER, the Golgi, the vesicles, is called the endomembrane system, and it's absolutely crucial for how the cell works and communicates.
Okay, we've talked about manufacturing and shipping. Now let's switch gears and talk about power. Specifically, let's look at plant cells, because they have this incredible organelle that we animal cells just don't get. Say hello to the chloroplast, the cell's very own solar panel. And inside, look at those little stacks of green coins. Each individual coin is a thylakoid, and a whole stack is called a granum. And it's right there, in the membrane of those thylakoids, that the real magic begins. So, what's photosynthesis? At its core, it's just this amazing process of turning sunlight into food. The plant takes in stuff we breathe out, carbon dioxide, plus some water.
Then, using the energy from sunlight, it smashes them together to create sugar, which is its food. And lucky for us, it spits out oxygen as a byproduct.
I mean, it's an incredible trick, right? But how? How does a tiny green disk inside a cell actually capture the energy from the sun, which is millions of miles away? How does that even work?
Okay, let's follow the energy. First, a photon of light zaps a complex called photosystem 2. This energy gets an electron all excited and is powerful enough to split a water molecule. That's where the oxygen comes from. Now, this high-energy electron gets passed down a chain, kind of like a hot potato. And as it moves, it powers these pumps that create a big pile of protons. That proton pile is then used to make ATP, the cell's main energy money. But wait, there's more. The electron then gets another power boost from light at photosystem I and is used to make another energy molecule, NADPH. So you end up with two batteries, ATP and NADPH, which are the fuel for the next part of the process, actually building the sugar.
All right, we've covered a ton of ground. So let's wrap this up with one final exam-style question to see if you've got it. Here it is, your final challenge. Imagine a cell whose main job is to crank out massive amounts of steroid hormones. Now, steroids are lipids, so based on what we've talked about, which organelle would you expect to be huge in that cell, and why? I'll give you a little hint. The answer connects back to a part of the assembly line that isn't the rough ER. Think about it. Thanks for joining this lightning review. Now go ace that exam.